or ethnicity that is spared this devastating disease. So we're going to kind of walk through that today and learn a little bit about where the field is currently. I do have a few disclosures, contract research, and I am working on a study currently with the Altec company that I'll speak on as well. So at the Center on Aging, we really are part of the global initiative to eradicate, not the Zika virus, but Alzheimer's disease from the face of the earth. And we're doing that in several different ways. We frequently hear in the media, another drug fails, and sometimes all people can see is the failures that we've achieved, but not the successes. With each failure comes a valuable learning lesson. And part of what we've begun to put together is such a clear picture of the disease and its progression, what's happening genetically, molecularly, biologically, leading to the eventual memory and thinking problems that one develops. And that is a pathway over time. And the field has really changed from thinking about Alzheimer's as that person who is struggling to get dressed in the morning, who has already suffered irreversible brain damage, to a disease process that is actually occurring over decades with individual targets for stopping this disease every step of the way. So at the Center on Aging, we are working literally with dozens of different agents for those that are normal but at risk for Alzheimer's, different agents for those that may have more advanced stages of disease. So an understanding of this process is critically important. Now, we think of Alzheimer's as a disease that affects memory and thinking, maybe behavior at times as well. And in the past, it's been largely thought of as almost a psychological disease, as if somebody, has a life experience that causes them to spiral downward cognitively. This is a medical problem. It's a biological problem. And we've understood this since 1906 when Alzheimer published his first monograph on a young patient, August D., age 51, looking under the microscope at her brain after she passed. He saw two characteristic features of disease. On the left-hand side of the screen, the neurofibrillary tangle. And this is an abnormal aggregation of what would otherwise be a normal intracellular protein forming neurofibrillary tangles. These are quite toxic and disruptive to the cell, eventually leading to cell death. And on the right-hand side of the screen, in a low-power microscope, we see the characteristic feature of amyloid plaques in the brain. Here, a nerve cell would be a tiny little speck like this. So these are monstrous plaques, even though here microscopic anyway. And they are composed of toxic proteins killing cells really from the outside in. And the diagnosis of Alzheimer's requires both of these to be present in order for the destruction of the brain associated with Alzheimer's disease to ensue. Up until about the past 10 years, the only way we've been able to positively confirm this diagnosis is at autopsy. By looking at brain tissue and seeing these two features, we could eventually say, aha, it really was Alzheimer's disease. But technology has changed, and it's changed tremendously. We've been able to piece together, and we're going to walk through this pathway that occurs over time from normal to Alzheimer's disease, beginning with the production of amyloid and amyloid plaques, leading further to neurofibrillary tangles and cell death. The brain begins to malfunction, to shrink. Eventually, when enough damage has ensued, we see memory loss. And when the memory loss is bad enough, we'll see the functional decline, the difficulty with daily activities that's come to characterize the Alzheimer's state. Again, up until the past several years, our focus has solely been here. We have ignored all of this. And yet we know from other diseases like cancer, that the earlier you get to it, the more effective your treatments will be, the higher likelihood for a cure. And that same strategy is really moving forward in the field. It's helped us to begin to redesign our diagnostic criteria. Classically, we have only considered this dementia phase, characterized by amyloid plaques, neurofibrillary tangles, perhaps brain atrophy on scanning, cognitive changes, and functional impairment. But kicking this back in 1999, the diagnosis of mild cognitive impairment, a prodromal phase of disease, very mild memory problems, but associated with the pathologic features. And as discoveries have progressed, we now are able to identify a preclinical phase of disease. People who are completely normal in their memory and thinking, and yet already have this pathology accumulating in the brain. Let's take a look at this and see how this works. I'm going to use the example of Ronald Reagan. I think this is a very effective way to walk through this. We all know Reagan was one of our greatest presidents. He suffered from Alzheimer's disease. Here we have the tabloids saying that he hid his Alzheimer's, or Nancy hid his Alzheimer's from the president for 5 years. I don't know if that's true or not anyway, but it's interesting that Reagan's life kind of paralleled the major discoveries in the field of Alzheimer's disease. So we can look back at 1982. This is the first year that Reagan graced the cover of Time magazine. It's also true in 1982, there wasn't even a clinical diagnosis of Alzheimer's disease. For all intents and purposes, we'd see people, we called them senile, or sometimes referred to it as hardening of the arteries, or other things that really demonstrated our lack of understanding. And yet, work had been done suggesting that Alzheimer's is the #1 cause of dementia in the world. So scientists had been working on this, but really in the early stages of development, scientific progress is slow. About this time, work really started to ramp up. And in 1984, in the beginning of President Reagan's first presidency, amyloid was actually discovered as the component of these plaques that Alzheimer himself had discovered in 1906. So we began to understand the molecular makeup of these plaques. At the same time, the National Institutes on Health recognized that this was an impending epidemic, a major problem for the United States in the future. And in 1984, they decided that we needed a series of centers of excellence in the nation to begin to address this problem. They set up the Alzheimer Disease Center program, 10 centers originally across the United States to battle this disease. The University of Kentucky, I can proudly say, was one of the original 10 centers, and we've remained continuously funded in this effort ever since as one of the leading centers in the world. It's also at the same time in 1984 that the first clinical criteria was developed for Alzheimer's disease, mentioning memory problems spread to other areas of the brain that would lead to other difficulties, perhaps difficulties with language, difficulties with visual function, difficulties with higher order thought processing, and importantly, ruling out other medical causes for the disease. This began to allow us to begin to collect cases nationally, so that we could begin to share information, not just on the biology of disease, but also on the clinical picture of disease. So this is the first discovery, and it's interesting how history parallels actually the disease process itself. We first discovered information about amyloid plaques. And we've come a long way since then. In 2005, there was a discovery coming out of the University of Pittsburgh, the Pittsburgh Compound B, using PET scanning, which is a routine medical technique frequently used to identify cancers in a very early stage or metastases, cancers that cannot be seen by conventional MRI or CAT scan. That uses radioactive sugar. What the researchers here did was they coupled that radioactivity to a compound used previously in pathology labs to stain the plaques in the brain. And we can inject this in people's veins. It will circulate through the body, and if they do have amyloid plaques in the brain, it will pick them up. So here is a normal control, somebody with normal memory and thinking, and they do not collect any of the radioactivity in the brain. Here, we have a case of Alzheimer's disease, and it lights up in pink and red. This is amyloid plaques in the brain, for the first time in living human beings, visualized without the need for an autopsy. When we actually compare this imaging to what the pathologist sees at death, it has an almost perfect one-to-one correspondence. This is exactly what the pathologist sees. We were surprised at the time, and the work that we've done over the last decade or so, that many normals also seem to show this pattern. And that was the beginning of unlocking some of these mysteries. So if we look at the prevalence of Alzheimer's, the number of people that have Alzheimer's by age, what we see is after about the age of 65, this begins to increase, and it increases exponentially. Some population estimates, as many as 50% of those over the age of 85 have Alzheimer's disease. So the frequency can actually be that high in the population. This is clinical Alzheimer's, people actually having trouble with their memory and thinking, having difficulty with their daily activities. Now, we also know from autopsy studies over the years, people that may have passed away from a heart attack or unfortunately hit by a car trying to cross Broadway. Wait for the light, please. And when they've come to autopsy, we can measure plaques. Now, fascinating. We see those plaques actually begin to exponentially increase at about the age of 45. Now, if that's not a frightening thought to you in this room, you're a lot younger than I think you are. Because it's frightening to me that this could actually be happening in my brain right now. So fascinating, these blue dots are what this PET imaging shows us now. It shows us again exactly what the pathology shows us. And this tells us that the disease is actually beginning 10 to 20 years before the first memory problem appears. That's a long time. Perhaps to address the disease of Alzheimer's, we should not be thinking about those who are 75 or 80. Perhaps we should be thinking about those of us that are in mid-age, in our 40s and 50s. And this is a fundamental discovery in the field. It also, and I am an eternal optimist, is a silver lining in the cloud. That means we have time, right? It's very, very different. We don't have time when somebody already has had major destruction of their brain. We've run out of time. Here we have time to intervene, and that's incredible. So we've worked over the years. If we understand that this amyloid increase is the first thing happening in Alzheimer's disease, the strategy was to attack that Alzheimer's disease. And many of you may not be aware, but there actually was an Alzheimer vaccine created in 1998. And this is mouse Alzheimer's disease, what we call Mouseheimer's disease. And this mouse did not receive the vaccine. By 18 months, all these little black dots are amyloid plaques in that mouse's brain, eventually going to destroy that mouse. An immunized mouse has their brain almost completely cleared of those amyloid plaques. So this was a real discovery that really launched the first human trials of this vaccine, and we were so excited. We thought back then as if Salk walked into his laboratory and on day one discovered the polio vaccine, not realizing there was a lot of work to be done. We did give this to humans, and it looked like it worked to clean amyloid out of their brains. But unfortunately, the vaccine itself seem to attack the brain. And what we see in white here is encephalitis, what was a fatal meningoencephalitis in about 6% of the folks that received this vaccine. That's 13 people. They died of this encephalitis. We could not turn the vaccine off afterwards. So an important discovery, but this really halted progress in the field at that moment with considerations of, yes, we can do this, but we really need to do this in a safer fashion. So we move to passive immunization, the administration of antibodies intravenously. And we've been working with a large number of agents over the past 10 years or so that are designed to do this. And here again is a patient who entered the study. And what you see in red are the amyloid plaques. After 18 months of receiving one of these antibody infusions on a monthly basis, the brain is almost completely devoid of amyloid. So really an amazing discovery, again, telling us that for the first time, this is not inevitable. We can change the disease process. Again, the study was done in people with end-stage Alzheimer's disease, people with late-stage disease, already with massive brain destruction. The disease did not stop because we removed amyloid at that time. And similarly, we saw evidence for inflammation of the brain. Now, this is a little more controllable with antibodies. We can administer them. If we start to see inflammation, we can stop, and then that will recover over time. So it gives us some control, but again, another major failure, putting us back to the drawing board. Well, it turns out over that time, we've been looking at amyloid, how it's processed in the brain, and it comes from something called the amyloid precursor protein, normal protein in all of us. And this process actually is ongoing in all of us. It's ongoing in our children, in our grandchildren. All human beings at every stage of life are producing some level of amyloid proteins that are Alzheimer proteins. Normally, we produce them at such a low level and get rid of them so efficiently, they never build up in the brain, they never lead to disease. In Alzheimer's, this protein is cleaved by an enzyme called beta secretase and then further cleaved by an enzyme called gamma secretase, leading to this isolated fragment, half of which is very lipophilic, fat-loving. It sits within the membrane, and half of it is very water-loving. It lives in the extracellular environment. Well, that fat portion from the membrane does not like to be floating around in solution. It wants to aggregate, just like oil and water don't mix. These start to bind together to form soluble oligomers, which are highly toxic to nerve cells, and eventually are deposited as plaques. So this really has enabled us, and the work over the last 10 years has been to parcel out. Do we want antibodies that attack the plaque? Do we want antibodies that attack just the oligomers? Do we want to generate antibodies that affect just these monomers? Do we want to inhibit beta secretases or gamma secretases? And if we are going to use these strategies, when in the disease course do we want to use them? And so right now, the major initiatives globally, actually initiated here in the United States, are trials of preclinical Alzheimer's disease, what we call the A4 study, where we're giving these antibodies to people who are still normal but have those positive PET scans. So these are people who are still in that preclinical phase of disease, like finding atypical cells in a pap smear and getting rid of it before you develop cervical cancer, a small bit of calcification on a mammography, and getting rid of it before you have breast cancer. This is the effort currently. The field is also working on safer vaccines. These are just starting to come out into human trials over the last year or two. We don't yet have any data. We do know that the preliminary studies suggest that what we've learned over the last 2 decades since the original vaccine trial is highly promising, that we can generate a safe vaccine. And it'd be really nice if we could send our children in, not just for their MMR, but for their MMRAD, and know that they would not have to suffer from this disease in the future. Or, like the varicella vaccine, we could also get it as adults, right? And potentially prevent that. So the field is moving forward. We're looking at different agents. We've begun to look at, in real earnest, BACE inhibitors, beta secretase inhibitors, really preventing this process of releasing amyloid. That actually forces the amyloid precursor protein into what we call a non-amyloidogenic pathway. This is a brain health pathway, if we can push everything in that direction. This is an Alzheimer pathway, what we'd like to prevent. And given these agents to people, we can see dramatic reductions in their spinal fluid amyloid production and an increase in the brain health promoting fragments, even over a period as short as 14 days. So these are being produced by several companies out there. I show you here the Lilly compound, but Merck has an agent like this, Novartis has an agent like this, several other companies working on this as a strategy. So let's get back to Alzheimer's disease. We've jumped now from 1982 and early discoveries in 84 that have taught us about the amyloid part of the disease to 1986. Reagan again graced the cover of Time magazine. And this is a period of time where another series of breakthroughs were instrumental in guiding the field and teaching us about the disease. It was 1986 when tau was discovered as the protein involved in tangle formation. Tau protein is normally in all of our cells. It helps our cells maintain their structure, maintain their length, and then extend these processes out so that one nerve cell can talk to another. In Alzheimer's disease, that tau is disrupted. It falls off the microtubules, the skeleton of the nerve cell collapses into one of these neurofibrillary tangles and dies. And it was also 1986 when those original plans for a United States national Alzheimer's Center program was initiated to move worldwide. And so the consortium to establish a registry for Alzheimer's disease, what we called CERAD, was established. The first effort to get scientists across the globe to start talking with one another, to start using the same diagnosis, the same treatment to cure disease. So we've moved into this phase of disease. And we've learned a tremendous amount here. We've learned from animal models. It's a fascinating experiment. These animals produce abnormal human tau protein only in the entorhinal cortex where the disease starts. And after 12 months, 18 months, 21 months, 24 months, we see the abnormal tau protein moving from where it originally was and spreading throughout the brain, nerve cell by nerve cell. This is a process that once started, presumably by the amyloid, continues to perpetuate. We've also developed ways, this is still experimental, to utilize PET scans to identify neurofibrillary tangles in living Alzheimer patients. So now, what Alzheimer's could only do with autopsy in 1906, now we can look at both plaques and tangles in living human beings. And the efforts have begun to stop this tangle process as well. We can see in animal models, many tangles, animal models of Alzheimer's. We can treat them with antibodies to remove this tau protein, and we can virtually eradicate the disease. We've begun to look at this in humans, and this is human data with one of the first monoclonal antibodies against tangles. And we can see that we can dramatically reduce the level of tangles, and concordantly, a measure of dementia drops. The patients improve with this treatment. So this is just moving into phase 2 trials now, very, very important. Moving forward, we have the plaques and the tangles in the brain. We're going to start to see the brain malfunction. We can start to see abnormalities in sugar PET scanning. And these are shown here, a normal brain lighting up with a FDG PET, mild cognitive impairment, mild memory problems, brain turning down. Alzheimer's, we've lost a lot of activity here. This is a real problem. And so unlocking why the brain is turning down metabolically, in addition to the overt cell death, is very, very important. Many researchers have begun to think of Alzheimer's as a type 3 diabetes, a diabetes of the brain. And what we do know is that if insulin is reduced in the body, insulin degrading enzyme is reduced, and we do not break down or destroy the amyloid in the brain. So this is really, really important. And trials of intranasal insulin that won't affect blood sugar have been developed, and they can show dramatic improvements in memory and thinking. Here, the reduction in score is an improvement as well, and here the preservation of scores is an improvement over time. Looking at imaging of the brain, all of these red areas are areas that were turning off in an Alzheimer patient that turn back on to normal levels with the administration of intranasal insulin. This is a major national study ongoing currently. We're looking further at downstream pathways, RAGE inhibition, the receptor for advanced glycation end products, a marker of the age of nerve cells that triggers their death and removal from the system. RAGE inhibitors are intrinsically involved in the amyloid pathway, and we actually are working on this currently. There was a study that was conducted several years ago, 2011. The study actually was stopped early because of a futility analysis. It looked like it was going to fail. At the end of the day, the study showed statistically significant reduction in decline in Alzheimer patients, teaching us that we can target these metabolic cell destruction pathways. And then, of course, if that brain continues without treatment, it will begin to shrink. And we'll see that on MRI scans. At the doctor's office, we can see this 5 years before the first memory problem appears. We can begin to see these areas in red and yellow shrink beyond what's expected for normal aging. Saw a young lady in clinic yesterday, 56 years old, strong family history of Alzheimer's. She tests completely normal. She already is demonstrating this shrinkage. We are working with her to get her on appropriate medicines. The medicines that we may use at this stage are again going to be entirely different. This is a study of a fin kinase inhibitor. It's an anti-cancer agent repurposed for Alzheimer's disease. And it prevents the amyloid process, which triggers neurofibrillary tangles. It cuts this off right here. So we can have amyloid in the brain, but it's not going to destroy the nerve cells. The data is quite convincing. This is a project that was begun at Yale University by my close colleagues, Dr. Strittmatter and Van Dyke, and currently is in phase 2 trials across the United States. We've begun to look at genetic vectors, implanting adenovirus that produces some of the molecules that are lost in Alzheimer's disease to try to restore brain health. Very early studies, just a few patients in each group, beginning to show that high doses of this medicine, I'm sorry, high doses up in blue, actually preserve memory and thinking and preserve nerve cells in the brain that would be lost otherwise. So 1990, and I know we only have a few minutes left, we're going to move through this, the onset of MCI. 1990, that term was first introduced. We began to unlock the genetics, autosomal dominant and risk factor genes for Alzheimer's. This is also a notable year I began researching Alzheimer's, and the field slowed down tremendously. So mild cognitive impairment developed as a predictor. If we look at people with mild cognitive impairment, they progress to Alzheimer's at about the rate of 10 to 20% per year, whereas the normal elderly population, 1 to 2% per year. So Alzheimer's, or Reagan himself, in 1994 addressed the nation. He was out of office at that time. He confided that he had Alzheimer's disease and really began to raise further awareness that this is a problem that we can share and we can increase awareness by doing this. I really applaud President Reagan and Nancy for coming out with that. Eventually, the disease progresses to the point of functional loss. We're working on this as well. We haven't forgot about the Alzheimer patients. There are new agents that are coming out targeting different receptors in the brain to try to improve function, even if we've had that kind of cell death. This is data from a class of new drugs that hopefully we will hear about in late December of this year in terms of whether or not they're efficacious, potentially bringing new medicines onto market early next year for symptomatic treatment, showing significant benefits for the treatment of Alzheimer's. 2001, 7 years into AD, President Reagan does not know who this young lady is. He cannot tell you her name. He loves her dearly. He does know that. In Alzheimer's disease, we've seen that process. Amyloid to tangles, to cell loss, to brain atrophy, to memory problems, to functional decline, lead us eventually to the fact or the ultimate outcome of a destroyed brain. And in 2004, President Reagan passed away. So, as I always like to say, we know the answer to the mystery of, did Reagan have Alzheimer's when he was a president? We used to kid Ron Peterson. He was my mentor at the Mayo Clinic before I came here. He was President Reagan's doctor, and we always elbowed him. Come on, Ron, tell me, did he have Alzheimer's? And of course, doctors can't share that information. He would just smile and say, Greg, you know I can't tell you about that. So here's Reagan's presidency timeline. We can look at this and learn about the disease. 2004, he passed away. Alzheimer's has a typical 10-year course. That brings us back to 1994 when he was diagnosed with AD. Mild cognitive impairment, early memory problems preceding Alzheimer's has a typical 4-year course. That brings us back to 1990. We can anticipate that his mild cognitive impairment began just as he was leaving the White House. That means that in 1979, years before Reagan took office, he already had amyloid plaques in his brain. I would just raise, perhaps we should screen all of our presidents with an amyloid PET scan. Nonetheless, so that poses the question, did he have Alzheimer's when in office, or did he not? Clinically, clearly, he did not. Biologically, yes, indeed, Reagan had Alzheimer's disease. So what does that mean for the size of this problem? Well, typically we look at the population of the United States. This is 13% of the population, 39 million people over the age of 65. We blow that pie up and we say 13% of those have dementia. These are the figures you hear, 5.5 million people currently in the United States. But if we begin to include those with mild memory problems, those numbers rise. Now we're talking 29% of the US population over 65 years, 11.5 million. And the real size of the problem that we're working on is this. 1 out of 3 people with normal memory and thinking over the age of 65 already have plaques in their brain. And if we total those numbers, that means 21 million people. 2 out of every 3 persons over the age of 65, whether they have memory problems or not, is affected by this disease. And that is a big target. We need to find ways to do this. And vaccines may still be many years off. We are working with Alltech on a very interesting agent they've developed over the years. They've been using this as a nutritional supplement in animal husbandry. It turns out that this is a unique form of yeast selenium, very different from other forms of selenium, functions as a powerful antioxidant. It also seems to modulate gene expression that's unique amongst forms of yeast selenium. So you can't just go to the grocery store and pick up yeast selenium, or to the pharmacy. They're quite different one from another. Work done early on at the Sanders Brown Center on Aging on mice with Alzheimer's disease. This is, I'm sorry, I've flipped my signals here. This is a placebo, amyloid plaques in the brain, treated with AT001, almost completely clean. Reduce that amyloid by about 60%. Quite profound. It's very, very safe, and we took this with Alltech into phase 1 studies. 24 normal elderly. We were able to demonstrate a dose response titration in the blood, and measuring spinal fluid, we were able to show that AT001 makes it effectively into the brain. Interestingly, we did not see effects on oxidative stress. Instead, what we saw in a dose-dependent fashion was a reduction in prostaglandins, inflammation in the body. And looking at the spinal fluid, small number of patients, we saw a trend that didn't quite reach statistical significance for stopping the production of Alzheimer proteins in the brain. This was enough for us and enough for Alltech to move forward with what we call the Alpha study, AT001 for the long-term preservation of brain health and aging. This is funded by Alltech. We're now studying 60 patients over a 1-year period, bringing them in, measuring their brain by MRI, CSF, spinal fluid, blood draws, looking at this inflammation and change in Alzheimer proteins. We expect enrollment to be completed in the next few weeks for this, and for us to have results probably in the next year. I think that this is important, and this is another way that we are working to bring better medicines, potentially prevention, to the field of Alzheimer's. There are some key concepts we need to remember. There are millions, not just in the United States, but across the globe, millions, tens of millions, that are amyloid positive. Our treatment is going to need to be low cost. It has to have an excellent safety profile. This is one of the reasons why we're so intrigued about AT001. It is poised to be distributed at very low cost with an excellent safety profile, essentially across the globe. Preferably an oral agent. We're not going to be able to treat people all over the world with intravenous infusions. We should target the disease early for prevention. And then there's always a consideration we may need 1 or more treatments in combination. We do need to do this now. The medicines that are leading towards the cure we need by 2025 to keep the US solvent, and many other nations across the globe, needs to be done in that time. That gives us 7.5 years. That means that agent must already be in our hands. And we think the number of agents that we're using currently, AT001 and many of the others, are that path forward to, if not completely eradicating Alzheimer's, at least dropping that prevalence of disease to a manageable level while we refine our treatments and cures. And with that, I will stop. I know I probably went over a few minutes. I apologize for that. Cutting into your coffee break.